All right, good morning, everybody.
Good morning.
Oh, we've got a cat going off now. Oh, we can see my daughter. Kind of dim. My youngest. And my dog, obviously. Okay, so we've got quite a bit to get through today. So I'm going to waste no time. The reason I was a bit late on was because I was digging through an old paper of mine to use as an example later on. Okay, so we started off, not there, or we left off rather, around here talking about epigenetics, right? And this is a pretty huge and complex subject, far more complex than I truly understand, to be honest, right? So this isn't really my field of expertise by any shake. But essentially, there's a kind of a balance between, hey, quit it. Um, Between the dynamic changes in chromatin that are going on in any given cell at any time, and also the long-term essentially shutting down of transcription of particular programs or things that shouldn't be expressed because it would be inappropriate to do so, right? And by inappropriate, I mean, once you've developed something, you don't need any more of it. It would be inappropriate to start that program up again. And in many ways, cancer development is the restarting of those inappropriate programs of development, essentially, but out of context. And so um, the kind of day-to-day stuff is really carried out by all of these different approaches. Um, but also this kind of these chromatin remodeling complexes are actually involved a lot in the specific areas of DNA, opening them up for transcription. Whereas these chemical modifications of both histones and DNA can be both. It can be small areas, it can be large areas, it can be totally shutting down transcription. And actually, in fact, again, ladies seem to get like the special attention when it comes to genetics for various reasons. But in females, in women, or any females actually, to be honest, any female that has two X chromosomes, right, in every one of the cells of that organism, woman, if you want to talk about humans, but it applies for non-humans as well. One of those X chromosomes is shut down, right? And that's to make the gene dosage the same for the X chromosome between males and females, because males only have one, right? So, and that's done in a mosaic way. Actually, that's the reason why you get, oh, dang it, the words left my memory. Those funky cats, female cats that are black, orange, and white. Dang it, what's the name of that? Huh? No, not tabbies. That's the type of patterning. Calicos, that's it. Getting help from my wife in the kitchen. Um, yeah, so they're able to have three different colors from two genes because of X inactivation. That's why you can't get a calico male cat. So if anyone tries to sell you one, like a calico cat that has testicles, they've been using some spray paint. It's a fake. Anyway, just take that from me, like a good, useful life tip for my students. So chromatin remodeling complexes, essentially the aim here is to expose DNA or not, as the case may be. They can also be negative, either expose or hide DNA from transcriptional machinery, right? And yeah, essentially the idea is that some of the DNA that transcriptional machinery needs is hidden. I mean, this is actually out in plain sight, but it's not accessible. And the reason why it isn't accessible is it's wrapped tightly around proteins. And typically DNA binding proteins, they can actually kind of hit on the surface like that, but quite often they actually clamp around the DNA to bind it securely. And so if this DNA is not out in the open or loose, kind of like flowing off of the histones, it's essentially inaccessible, right? And typically the idea for chromatin is the default state is off, right, of transcription. That's generally true for eukaryotic genes anyway, in terms of the control of transcription. But in terms of chromatin, the default state is heterochromatin, right? So those genes are off. They have to be told, or that chromatin has to be actively changed to allow gene expression, right? And so one way we can kind of make this accessible is just to slide these histones along a little bit. We can rotate the histones so that the DNA that was on them is no longer on them, and then we could just kick them out and get rid of them, all of which takes ATP, right? So this is an energy intensive process. Obviously, it has to be done for a reason. And so, yeah. And also this kind of get back to this, kind of bouncing around a little bit. I want to make sure I get the whole picture across. Essentially, modifications that are made to the histones themselves act as cues for their own remodeling, right? So even though chromatin remodeling complexes move the histones themselves, they physically move them along, around, or off, they require a signal to do that, and that signal is actually part of the histone, right? So they recognize that, use that to determine which histones they're going to remodel or which complexes they'll remodel. So there's a lot of interplay here. There's a lot of talk between different parts of the system, the DNA, the histones themselves, and the proteins that modify the DNA and the histones. And there's talk in all directions in there. So this is we don't have a good diagram of sliding, but you can kind of think about how that would work. But using that same picture back up here, right? Here's our transcriptional activator protein binding site. And so this is typically the bit of DNA that's recognized by one of the first proteins that assembles to begin transcription. We've got our tartar box binding protein binding site, which is a little bit cumbersome. And this binds, obviously, the tartar box binding protein, or is recognized by, more accurately. And that is what brings in the RNA polymerase holoenzyme to begin transcription. Right, so both of these have to be active, and they typically are spaced apart in such a way that when they're both accessible or they're both inaccessible, right? And so if we go back down to here, right, some of those histones must have been modified in a way which recruits this chromatin remodeling complex, right? And it's a large complex of different proteins, right, which do different things. And essentially what they do is if you imagine that DNA is literally wound around these histone complexes, right? These 8 histones at the core. If you rotate that, then the DNA that's wound around it is going to change, right? It's literally like having a kind of something wrapped around your finger and like just rotating your finger around or your hand around. What's wrapped around it will change as a result. And so the idea here is that these histones are rotated along. And again, this is why this linker DNA is important, because it allows that to happen. If these nucleosomes were stacked really close together, it would be very hard to do this, right? So this kind of gives you some wriggle room, essentially. And these are rotated, and now the binding sites are exposed. So now we have our TAP binding site, we have our TBP binding site. Those are both now in free air, essentially. And remember, this is in a nucleus that's jam-packed with DNA and proteins and other bits and pieces that are going on. So in that mix will be transcriptional activator proteins, title box binding proteins, RNA polymerase complexes, transcription factors. All of those proteins are going to be kind of in that mix. And so just by stoichiometry, at some point, that TAP protein will go, oh, look, that's something I recognize, and it will bind to it. And now that's a big thing kind of like flapping around in the out off of that bit of DNA, and that's now recognized by the Tatabox binding protein, right? So that comes in complexes. And out there's an even bigger complex, which hitting the fake plant behind me, which is RNA polymerase. And now that sees something, it's like, okay, that's something I recognize. And so the whole process, and it's a very large complex of proteins, of many, many proteins that comes together. Those all find each other because now they're in a kind of format or a situation where they can assemble together. And then as soon as that happens, the RNA polymerase comes in, and the RNA polymerase is actually pretty good at kicking histones off of DNA itself. It's kind of like a snowplow. Just kind of goes along, or a train with a plow in the front, like pushing cows to either side and stuff like that. It actually is able to get at the DNA even if it's wrapped around histones. So the key point is not the RNA polymerase binding in terms of histones, it's making sure that the binding sites for the proteins that help it bind DNA are accessible. Because as soon as those become accessible, the complexes start assembling, and then RNA polymerase is brought in. And as long as various other things are present, enhancers and transcription factors and the like, RNA polymerase will bind and transcription will be initiated. Right, so now that DNA is effectively open for transcription. Now, obviously, if you want to turn this off, a very easy or straightforward way, rather, you don't have to change that mix of proteins that's in the nucleus. All you do is have to turn those histones back again, and that DNA becomes inaccessible, and those proteins kind of floating around, jostling around in that mass, no longer recognize it, no transcription. Because RNA polymerase cannot recognize and bind DNA on its own. It has to be helped to do so. Right, and obviously a very even more straightforward way of kicking this whole thing into gear is just kicking the histones off. Right, and so again, this requires ATP, and now rather than kind of just shuffling things around a bit, now you can physically just remove a histone octamer or two from the DNA. And so all of the DNA that was wrapped around that histone core particle now is free, right? And so that's not just a little bit, that's actually a fairly decent sized chunk. That's probably, like 300 base pairs in total, give or take. The DNA that was wrapped around the core plus the two lots of linker DNA on either side. And so now that really opens things up, right? This is actually a fairly kind of serious process. This actually happens during DNA replication too, right? To allow the DNA polymerase and associated machinery to copy DNA, right? But in transcription, you can actually kick things off very quickly. And so you can see how there are extra subtleties going on here, right? Just doing this is going to make this available, but not like super available. Doing this is going to free up an entire piece of DNA and make it very reliable for transcription to start, because now there's nothing impeding the access of those proteins to that DNA. And again, this is a very dynamic process. It's constantly shuttling between one form and another, right? It's not
So
something that's just done once and left.
does the other one use ATP or doesn't use ATP?
Yes, it does use ATP too.
So both will use, but the one that kicks out the histone is going to use a lot more.
I don't actually know the relative amounts, because in some ways, like moving things around is actually harder if you think about it mechanically. I don't actually know. But this is a much more kind of definitive, I guess, a much more reliable way of making DNA available for transcription. I don't know the relative energetic cost though. Another cat is going off. Okay, so the rest of it, chemical modifications of proteins and DNA, is arguably the most complex part, especially of histones, of epigenetics. And it's basically centers around two things: methylation or demethylation, and acetylation and deacetylation. Right, so those are mirror images of each other. So methylation, that's the addition of a CHC group, and obviously demethylation, if I can spell that. And methylation of DNA is repressive. And methylation of histones is usually, but not always, repressive too. And yeah, it's just complex. I don't know of any examples where histone methylation activates transcription, but that doesn't mean that they don't exist. Just that I haven't come across specific examples yet. Now, acetylation only of histones, right? You don't get acetylation of DNA, is generally activates transcription. Ooh, let's actually got to be careful about that. Allows activation of transcription. And essentially for DNA, only cytosines are, I spelled that wrong? No, I think I spelled that right. For histones, generally, again, I got to be careful about being too specific. It's the lysines in the histone tails are acetylated or methylated. But also there are a lot of lysines in, how many different types? There's 4 different types of histones. So you can presumably methylate one of those lysines and acetylate another one and you'll get some different combination of effect on chromatin. And so I want to also give you some names of things, like what does what. Aha, gotcha. We have DNA methyl transferases and demethylases. Those add and remove methyl groups. And we have histone acetyl, if I can spell that right. There we go, that was a problem. Transferases, histone, come on. Deacetylases. And those add, remove are, what's methyl? CH2, CH3 groups. So HATs and HDACs are kind of how they're referred to. And DNMTs and DMs, I guess. I don't actually talk about those much, to be honest. But HDACs are super important. Think of it like HVAC, you know, what keeps you warm in winter and cold in summer. And so there are a lot of different kind of little switches you can flick on and off by acetylation and methylation of histones. And so really it's the kind of the sum of those changes which affect how tightly bound DNA is to histones. But generally speaking of histones, acetylation will make DNA more available for transcription. Deacetylation will reduce that. And methylation generally reduces the availability of DNA for activation. These things also act as cues for other proteins too. So the chromatin remodeling complexes, for example, detect the methylation and acetylation state of histones, right? There are proteins that recognize that and then methylate DNA, and vice versa, proteins that recognize methyl groups on DNA and then change the activity state of the histones, right? So there's a lot of interplay and crosstalk between all of these different players in chromatin maintenance and remodeling. But the key points to remember are these two different groups, right? What adds them? What removes them? What's the consequence of the adding or removing of that? Okay. Oh, and also, obviously, there are histone methyltransferases and demethylases, not just DNA ones. Forgot to put that in there. So as always, this chat thing gets posted in Blackboard. So if you want to just double check on that, as a kind of a supplement to the notes in the PowerPoint, that's not a bad idea. Okay, so essentially this is like the kind of big picture look at things, right? We've got our DNA wrapped around histones, and we've got 4 pairs of histones, right? 8 in total. Each of those histones has these long tails coming out, right? These tails are what are modified. They also are what vary the tightness by which this DNA is wrapped around the histones, as well as a whole bunch of other stuff, right? And so typically, if these tails are methylated and deacetylated, DNA will be bound very tightly, and those nucleosomes will be packed together very tightly. If you demethylate and acetylate those tails, now those histones spread further apart, or rather the nucleosomes do, and the DNA kind of relaxes off of those histones. Because the big deal is, oh, here you go. Here's a question. I asked this of the class yesterday. Okay, so you know what charge the backbone of DNA have? Oh, actually, quick, change that. Hopefully no one saw that slide. Right, what's the charge of DNA?
Negatively charged.
Correct. So if lysines are the amino acid residues that are modified to vary how tightly these tails bind to DNA, what charge naturally do you think these lysine residues have? If DNA is negative, you can't change the charge of DNA. It's inherent, right? So if you want to change how tightly these tails bind to DNA or not, what charge are you going to change? What's attracted to negative?
But what was like what the lysine? What is that role play? I got confused with that.
So, oh, thank you, Levia. I just saw that. Yes, that's right. So lysine residues, and they're called residues because they are amino acids, but they're amino acids in a chain, right? So it's like saying, A, C's, and T's and G's are the bases, right? Because the backbone is the same. It's the same for proteins. So lysine residues are found in the tails of histone proteins. And lysine residues in the tails are or can be acetylated or methylated. So if we're messing around with lysine residues and DNA is negatively charged, what charge normally do you think lysine residues have? If you want to make something stick to something negative, the best way is to make that something
Positive.
positive, right? If you don't want that thing to stick to something negative, you can turn that positive thing neutral. And that's what acetylation does to a lysine. It neutralizes its positive charge. And so because, get you on the next one, because those positively charged tails are now no longer positively charged, they're essentially neutral or less positively charged. Those tails don't bind to that DNA as tightly anymore. And if they're not like kind of tightly kind of grasping that DNA now can sort of relax off of the nucleosome, and now it's much more accessible to proteins that bind to that DNA that can activate transcription or begin the process of recruiting different parts of the transcription machinery. So flipping back and forth between those, we don't have to change the DNA. All we have to do is change the charge state of the histone proteins, or specifically the lysines in the tails of the histone proteins. Yeah, exactly, Ali, that's right. So if you weaken this interaction, that DNA becomes more accessible, it becomes more likely for proteins to bind it. And then like transcriptional activator proteins start recruiting the other stuff. So essentially, by adding something to the histones, you can essentially like turn off that magnet, right, which attracts the DNA to the histones. And so acetylation, even though it's something that changes the histones, is positive for transcription because the DNA is more accessible. Removing those acetyl groups and actually replacing those acetyl groups with methyl groups does the opposite. I don't actually, to be honest, really understand the mechanism behind histone methylation, like how that affects things. So it's kind of hard for me to describe that if I don't understand it myself. But if you instead methylate those histones, A, you can't acetylate them, and B, you don't affect their charge. So you now have positively charged histones. You turn the magnet on, that DNA is tightly bound, and it can't be loosened. Right, so that essentially represses transcription of that particular bit of DNA. You're welcome. So essentially, it's just, kind of modifying how tightly bound that DNA is. And there's different ways in which you can do it. But essentially, what you're doing is changing the charge of the protein. And these tags, right? So these are called epigenetic marks or tags. These are also heritable. So every time you replicate your DNA or generate gametes as part of that, half of the DNA that you make is going to be have histone proteins that are tagged. Pardon me. So these tags are heritable, right? And this is essentially what we call the histone code hypothesis, right? So these tags are both heritable, even though they're not DNA based. They don't change the sequence of your DNA, but they do alter how that DNA can be used. So even if you have exactly the same DNA as someone else, if your histones and DNA are modified by acetylation and methylation, the pattern of expression of those genes will differ between those two individuals, even though they have exactly the same instruction set. And so it's not DNA that does stuff, it's proteins that do things typically. And so if you change what proteins are produced from that DNA in terms of where, how much, and in response to what, you'll get a different outcome as an organism. You will look different, essentially. You'll function differently. No trouble. So the other one, which is kind of even odder in a sense, is DNA methylation, right? And so essentially what this is, here's our cytosine, this is our nitrogenous base, right? If you swap out this hydrogen for a methyl group, you change its properties quite a lot. and you change how it's recognized by other proteins too. So those DNA binding proteins will recognize a particular kind of look, essentially, of a DNA sequence. And so if you add a methyl group to those any Cs that are in that DNA sequence, you change how it looks, and you'll change how readily it's recognized by DNA binding proteins. And so typically these methylation marks tend to be in the promoter regions of a gene, right? They're actually based on, come on, get you, these CG dinucleotide repeats. So wherever you find C's and G's, and there's just two bases there, so they'll occur randomly pretty frequently, but they tend to occur more often in promoter regions. Anywhere you find these, they can be, they not necessarily are, but they can be methylated. And quite commonly in promoters, like the regulatory regions of genes, you'll find what are called CPG islands. So you'll find whole bunches of these C's and G's, or CG repeats. And actually, many moons ago when I was at a different institution, one of the students, see you, love, I supervised writing their thesis, did a project on Luna, come here, on a tumor suppressor gene called P70. There are a whole bunch of them. And essentially their role is to check up on the cell cycle checkpoints, right? So their role is to make sure that any cells that slip through the cell cycle checkpoints when they shouldn't kind of get shut down and die. So their job is really to kind of police the checkpoints, essentially, is kind of one way of looking at it. And there are a whole bunch of them. And what's often found in cancer is those tumor suppressor genes don't work. Or the proteins, there are fewer like transcription of those is lower. And so this student, can't actually remember his or her name actually, it was a long time ago, found that the promoter region of one of these tumor suppressor genes, P70, was heavily methylated in cancer patients, but not in individuals without cancer. So essentially something either that caused the tumor to grow or that was controlled by the tumor cells resulted in the downregulation of this tumor suppressor gene. And that downregulation, that kind of suppression, was done by the methylation of its promoter. And so essentially those transcriptional activator proteins, transcription factors, enhancers, all that kind of stuff, those proteins are less likely to bind. And also proteins that repress transcription are more likely to bind. So the methylations themselves don't, I mean, they do change the DNA, but they don't necessarily change the information. But they do change how that information is recognized by other proteins. So I always thought that was, even now, I think that's really interesting, right? So, you know, tumor formation is multifactorial, right? There are a lot of different things going on in it. So it's just kind of cool. I like talking about that stuff. And so also, just as a quick aside, this is DNA methylation is how the mismatch repair mechanisms that work after DNA replication is finished, how they recognize what is the old and correct strand and the new and changed strand. And so I don't know if you remember way back when we talked about this stuff, seems like a lifetime ago, right? But there's a gap after DNA replication where you can tell the difference between template DNA, the old stuff, and the new stuff, the daughter strands, right? And the reason you can do that is because the daughter strand hasn't yet been methylated, right, by these DNA methyltransferases. So once that does happen, any errors that were introduced during DNA replication become fixed, and they cannot be fixed. So that's kind of another aspect of this, which is interesting too. Kind of ties back into stuff that we learned some time ago. And so very quickly, I want to just give you a couple of examples of how these things can talk to each other, right? And in both directions as well. And so this is an example of a situation where you have a methyl group, right, attached to the DNA, but the histone conformation is fairly open, right, because it's acetylated. That tail doesn't bind to DNA. That DNA is accessible, right? So when that methyl group is methylated, that cytosine is methylated, you have that methyl group present. That's now recognized by another protein. That protein recruits HDAC, a histone deacetylase, removing that acetyl group and then promoting the addition of a methyl group in its place. Right, so the transcriptional silence in DNA basically positively reinforces that silencing by also modifying the histones near to it too, which further reinforces that silencing process. So these aren't kind of things that occur in isolation, right? The presence of one can lead to more of the presence of another and vice versa, right? And similarly, if you have methyl groups on histones, right? And so this is that histone code that I was talking about. This is histone 3 lysine 9, right? So if you ever read papers about that, and I kind of recommend you don't because they're really confusing. They will talk in this code, right? It's like another language, right? And so what they're telling you here is that of the 4 groups of histones, it's the H3, histone 3, which is being modified. And it's being modified at the 9th lysine, there's a lot of them, on its histone tail. Right, and so now we have information going the other way. Our histones are methylated. That recruits another protein, which recruits our DNA methyltransferase, and that now methylates the cytosines in those CPG residues. So it's not from DNA to histones. Now the information is going from histones to DNA. And so even though this is a fairly in-depth look at this process, because it's a very interesting process, you can also sense that this is barely scratching the surface too, right? Because this is just an astonishingly complex topic. Does anybody have any questions about epigenetics? Because we're going to move on from that to transcriptional activation. Nope. Okay, so now we've done the whole chromatin part. Now we're going to move on to the DNA itself and see how we can vary how frequently RNA polymerase is going to transcribe that DNA. And so to look at that, we really need to look at all of the different things that regulate that process, right? And there are a lot of them. Again, this is one of the many ways in which control of transcription is far, far more complex in eukaryotes than prokaryotes. Messier, I would say, less elegant, but much more complex, right? And so in eukaryotes, again, we have this kind of two two regions essentially, right? We have our core promoter. This is what's responsible for binding the RNA polymerase holoenzyme, right? So it contains the tau box. This is kind of in many ways the bare minimum that's required for transcription. So if you stuck just this, and it's, I don't know, kind of varies in size, right? I'd say probably 2 to 500 base pairs, give or take. If you stuck that in front of a GFP, a green fluorescent protein gene, you would see some expression from this promoter, right? Even in the absence of transcriptional activated proteins and other stuff, you would get some activity. Not a lot, and it wouldn't be controllable, but you would get some activity, right? So this is called the core promoter or the basal promoter or the minimum promoter or, various words to that effect, right? This is the smallest bit necessary, essentially, for this gene to be transcribed. Then you have a whole range of stuff, right? And this can be, whenever I've done this work and I've done a bunch of it, you usually grab the biggest bit of DNA that you can physically fit in front of whatever you're doing, because you don't know how big the promoter of a gene is. Typically, I'd usually, it's kind of, this is work that I did back in Penn State largely, between 2009 and 2014. I'll show you some of it in a second. But I'd try and get 2,000 base pairs, 3,000 base pairs, stuff, some about around that kind of range, right? As big as I could get, because I had absolutely no idea, and I still don't. It is almost not possible to know in many ways how much of that DNA is actually needed to control the transcription of that gene under all the different conditions. Because that bit of DNA contains all the different what we call cis regulatory elements, right? And these are bits of DNA that are recognized by proteins that either help or hinder the expression of that gene. But they can also, and this is an enhancer, right? So this is involved in the activation of transcription. This enhancer, this bit of DNA here, which is recognized by this protein, that could be millions of base pairs away. Technically speaking, it could be the other end of the chromosome. It's typically not on another chromosome. That would get way too complicated. But it doesn't have to be even within shouting distance of the gene. Even within, like, missile distance or plane distance, it can be a very, very long way away and still be very important in the control of the transcription of that gene. Actually, have you ever heard of polydactyly? Like when you have multiple extra fingers, they typically stick out of here. So it's kind of like that. We have multiple fingers. It's really cool. Super cool on cats. If you ever see a polydactyl cat, it's awesome. They've got these enormous paws. They're just like, they're vicious killers, basically, because nothing can escape them. But anyway, that's the result of a mutation in an enhancer, which is a couple of 100,000 base pairs away from the gene which controls the development of those of that fingers, the identity essentially of your fingers. Why you have a little pinky finger, and that pinky finger is very different from your thumb, basically, and why you have that pattern. It's very, 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 very tightly controlled in funky ways. But anyway, the enhancer that controls that is a very long way away from that gene. And so that's really the regulatory promoter, right? That's the region of DNA upstream of the transcription start site, which controls when, where, how much, in response to what, does RNA polymerase bind to that core promoter. And we've got a lot of different ones of these, right? So we have, and these are just some descriptions, right? We'll go through some examples of them in a bit. We've got enhancers, right? So those kind of increase the transcription or the rate of transcription. We've got silencers that do the opposite, right? These, and again, these are all pieces of DNA that are recognized by proteins. Essentially, a particular sequence of bases, which is recognized by another protein that comes by, or by a protein that comes in and binds to that sequence. Doggo. And these can overlap, right? So you can have a case where enhancers and silencers overlap. So repressor proteins, if they are bound to that bit of DNA, prevent the binding of enhancer proteins. You know, so it's extraordinarily complex, right? And really, these elements are what control where, when, and in response to what genes are expressed, right? Insulators are kind of funky. Essentially, their role is to prevent the activation of one gene spilling over into the activation of another gene next to it. Right, so if those two genes are involved in very different processes, you want to kind of wall them off from each other so that activating one doesn't activate the other one. And then we have these things called response elements. And essentially, these are, again, DNA sequences that are recognized by proteins, but those sequences are found typically in the regulatory promoters of genes which are involved in the same process. So if you think back to what we learned about bacterial control of transcription, their solution to the problem of having multiple genes involved in the same process is just stick them all together, right? And they're all controlled at once by one regulatory mechanism or mechanisms, right? Eukaryotes don't do that. So you can have a gene here on chromosome 1, you could have another gene on chromosome 22, you can have a gene over there on chromosome 5, and the products of those different genes are all required for a particular process, like gluconeogenesis, for example, or response to heat shock, right? So how do you control all of those different genes so they all produce what you need at the same time or in the same condition? And that's really done by these response elements, right? And so these coordinate or help coordinate expression of multiple genes in response to a particular stimulus. So if your body temperature starts going up, you're going to need lots of different things to help regulate that, right? You're going to have to produce things that promote sweating. You're going to have to produce proteins that help other proteins fold correctly or stop them unfolding, like you basically cooking, right? And so all of those different genes, even though they do very different things, you will find in their promoters the same bit of DNA that's recognized by a protein that's essentially becomes active when it gets too hot. And you can do that in multiple different cells as well, right? It's not just in the same cell, it's in multiple different cells too. And so in many ways, these are kind of distinct modules, right? So if you wanted to actually, I mean, you could create wing spots on moths if you wish to, right? You could take one of the elements that are responsible for pigment production in eyes and you stick it in front of a gene in that's expressed in the wings, for example, and you will get eye spots. Essentially, that's how eye spots have evolved in butterflies. It's by taking one or more of these elements, right? And somehow, we don't know how, that got stuck in front of a different gene, and then you get expression in a different place or in response to a different thing. And so you can get multiple copies of the same element. You can get a range of different elements, right, which will allow transcription in a range of different contexts. You can get elements which are very pro-transcription, elements which are against transcription, right? You can get a lot of different things of these. And essentially, the output depends on the overall content that you have in your regulatory promoter, and as well the proteins that are present in that cell. And so this is actually a viral promoter. SV40 is a virus. And this is actually a promoter that we use a lot to produce proteins for synthesis. So if, for example, you wanted to produce a lot of protein for something, I don't know what, maybe insulin, for example, to make a whole bunch of insulin protein to purify it and then to put it in vials for diabetics to use, right? You would stick insulin down here under control of this promoter. And then you would just get gangbusters amount of protein, right? Because viruses want to produce a lot of protein very quickly because they want to make more viruses so they can escape and make more viruses and so on and so forth. They're pretty single-minded when it comes to stuff. If they could, they're not even alive, so they don't have minds, but you get what I mean. Whereas other genes, for example, these, again, it depends what modules you have, depends on what is needed of that regulatory promoter, right? So histones, you always need histones, right? Particularly during DNA replication, because you need like the same number again of histones. So these are likely to be expressed, these genes are kind of a low and constant level, but need expression ramped up very quickly during DNA replication. Thymidine kinase, well, I don't even know what, well, kinase, so it adds phosphate groups to something. This may need to be expressed in response to something, but not expressed at all in other situations, right? And so essentially the combination that you have here is like a code, right, that allows you to unlock transcription under particular circumstances and at different levels of transcription too. And so if you wanted to, say, engineer this promoter so that it produces essentially transcription at a higher rate, even under the same context, you'd add more of these things. You just stick those in there. As long as you don't screw up anything else, the result will be you'll get a much higher rate of transcription. Now, if you wanted to turn down transcription, you could selectively mutate these GC boxes in this promoter, and that will dial things back down again, right? So if one wished to, you could actually use that information to manipulate that stuff. Okay, so now we've got, this is even funkier, right? So you can actually have multiple promoters. This is even weirder, right? And so I'm going to show you an example just because it's always fun to talk about something else, right? So here you go, it's my name, isn't that neat? So this was a gene that I did a lot of work on as a postdoc, And the product of this gene basically is involved in recycling the byproduct of NAD consumption. So NAD is involved in electron transport. Well, not electron transport, electron carrying between NAD and NAD plus, but it's NADH, sorry, but it's also used as stuff. And so this protein helps turn it back into more NAD essentially. Anyway, and so let's blow this up a little bit. Stop that. And so this is what the gene actually looks like, right? It has, and these lines correspond to introns. The dark boxes correspond to exons. And these little arrows correspond to promoters. And this thing here, this is a mutation which knocks out function, right? Basically results in the deletion of this last exon. And so this gene has 3 different promoters, right? And also this exon has a what's called a signal sequence, which directs it to be exported from the cell. So it's outside of the cells, right? And so you can have very different patterns of expression from each of these 3 promoters, right? So what does that look like, right? Because presumably the need for this protein varies in different cells. You can actually look up this paper and read it if you want. It'd be kind of fun. If you got some spare time, which obviously no one does. Right, and so when you use I think I talked about confocal microscope. We're talking about those neurons and keratinocytes and stuff. So these are confocal microscopy pictures of worms, C. elegans, you can see in here. They're expressing different constructs, right? And so basically, let's go back up here. You can take each of those promoters and stick a GFP down the other end of it, right? And so what we're going to have now is, at least in the case of these 3, a translational fusion between the protein, the PNC1 protein that I was working on, and something that glows. So essentially, wherever that protein is expressed, you'll get green. And for reasons that I honestly can't actually remember right now, because it's a long time ago. This was published in 2014. I also stuck the GFP just under control of that promoter, right? So these 2 proteins are the same, right? So the cDNA is basically all the mRNA turned back into DNA, right? Just makes it easier to handle with. So there are no introns in this. There's no processing. That's what these are for. But these proteins are the same, but the promoters are different, right? So this is 1B1 promoter, this is 1B2 promoter, right? So essentially what I did is I spent an awful lot of time taking pictures of worms and then an even more time trying to recognize what the hell those cells were. So at least for a little while, I got really good at figuring out what cells were which in C. elegans. Anyway, when you look at them under a confocal microscope, so actually these ones are confocals, these are different. What you see is the expression of those are in very, very different places, right? So this is the head of the worm. This is its intestine here. And these kind of bright dots, you can see here, these are the neurons in a worm's brain, right? It doesn't actually have a very big brain. It's called a nerve ring. Actually, Tanya, you laugh. 1/3 of the worm's cells are neurons. So think about us as humans, the relative proportion of our cell count, if we are able to count it, isn't as big. So they might be smaller, but they're not stupid. They're actually pretty cool when it comes to processing information. Anyway, so these blobs, right, these are pharyngeal muscles, but these blobs are actually neurons, right? And so I can't remember which promoter this is. I'd have to look at the figure legend. But you can see there's not very much expression of that particular version in neurons. And in this one, these pictures look really shitty because confocal microscopes take terrible white light pictures. That's why. It's not just because I was crap at doing it. These ones, now you can see a lot of expression in the intestine and some in the pharyngeal muscles up here. And then when you look at this one, it gets even more complex, right? So this is the same gene, right? The same gene, the same information. But depending on which version of that information is expressed from which promoter under control of different proteins, you get a very, very different output. And this is actually quite a neat picture. I really like this. This is taken on a DIC microscope, which is quite different. And so you don't get these super sharp pictures, but this kind of faint smoosh over here, this is actually protein that's in the extracellular space, right? So this is secreted protein, and it's filling in the gaps between, I think those are oocytes. So these are actually, no, not oocytes, these are embryos. That's a 2-cell embryo here, just there. So that's fun. That was a lot of work. But anyway, so just by having a different promoter in front of a different exon, you can get very, very, very different outcomes in terms of function, right? If it's under the control of this promoter up here, it's going to be a secreted protein. If it's under control of this one or this one, it's going to be non-secreted, right? It's going to only act in the cell in which it's produced. And why that exon is important, but it's not needed here, I don't actually know, right? So it's super random, right? So we have a better understanding of what's going on, but that doesn't necessarily mean we understand what's going on. But that's just a really good example of alternate promoters, right? How you can have the same set of information, essentially, more or less, but you can have a very different outcome in terms of function depending which of those promoters you use. And in a sense, this isn't technically an alternatively spliced gene because we're not leaving something out from a transcript in one transcript but not another. But you can see how we've got multiple different exons, but we don't necessarily use the same exon in every version of that protein. So that's one of the ways in which RNA processing can actually lead to kind of extra complexity, essentially. Has anybody got any questions about any of this stuff so far? We're not going to finish all of this today. We're going to do a little bit of on Tuesday and sacrifice a little bit of the biotechnology stuff because in a sense, they're already talking to you about biotechnology stuff anyway. Yes, Ali, that's right. Let me just check numbers. Yeah, that's right. So essentially 17, 18, 19, and 20. We're probably not going to do as much on 19 and 20, to be honest, because we're really going to have time for that only on Tuesday because the exam is next Thursday. But yeah, the information that we will be talking about will be from those PowerPoints. No worries. Yeah, it's going to move on past this, right? So this is another good example too, right? It's just that I wanted to show you some actual pictures of like what things look like. But the example here is not so much about where that gene is expressed, which is what I was looking at, but when it is expressed. And so you can have promoters which are active at different stages of development. So you'll get that protein produced in those different stages, and that protein may have a different purpose, pardon me, as a result as well. What else do we have? Oh yeah, we're definitely not going to finish this in the next 4 minutes. Okay. This funny stuff is just fun to talk about because you can actually it's just cool, right? I mean, this is really how we get such beautiful complexity, right? And what is it? Oh, I can't remember his name. Sean Carroll, that's it. Worked on the evolution and development of butterflies in particular. It's a very famous Evo Devo guy. Wrote a book called, I think it was Many Forms Most Beautiful. So that's a book worth reading. Essentially talking about how this complexity can come to be. So enhancers are much harder to pin down in terms of location and what even they are, right? Even even actually figuring out the DNA sequence of these things is extremely difficult to do. Right, I don't know in those promoters that I worked on, I don't know what bits of them are important. I don't know what things will recognize them or anything like that. Have no idea. That would be an enormous project. And so these are very hard to pin down, but essentially their role is to um, be recognized by proteins and to enhance transcription. Right, so the presence of those proteins is required for those enhancers to function in that particular way. And so now we have the case of, okay, it's not just what DNA sequences are present upstream of a gene, but also what proteins are present in that cell at that time that determine transcription of that gene. Okay, so now you have not just the information, but you also have the second part, which is, are there things around which will recognize it? Because if you think about this, that information is present in every cell that has that DNA, right? And all cells have the same DNA, pretty much. Red blood cells are an exception. They don't have any, right? But every cell has that set of enhancers upstream of that particular gene, but that gene isn't expressed in every cell type, right? So the determinant now is not just this bit, right? What are those bits of DNA there? It's also this bit. What proteins are present? And that's a very big part of the cell specificity of gene expression. Why different cells are different. And one of the reasons is that they have in them, as a result of other gene expression events, a different set of proteins. And so both of these genes are going to be present in both of these different cell types. Right? We've got a liver cell and a lens cell, right? And so lens cells, actually, this is super funky, and I hope I get time to describe it. But essentially, the cells of your lens in your eyes, there's a lot of cells there, right? And they're all clear. Because if they weren't clear, you wouldn't be able to see, right? You just go, oh, this is kind of like messy mush in front of my face, right? And so crystalline is a protein that actually clears those cells in your eye, in your lens of your eye. So at a certain point in development, the nuclei, the proteins and organelles and whatever kind of get cleaned out and replaced by this protein called crystalline, which essentially helps your lens kind of bend light to focus it on your retina, which is pretty cool. But obviously, you don't need that expressed in your liver. No one looks around with their liver, right? Because it's inside you, right? Livers do other stuff. And so in the liver cell, you don't have those genes present which will activate transcription of the crystalline gene. You don't have those proteins present. You do have proteins present that are required for expression of the albumin gene. Actually, I'm not 100% sure what albumin does. I know it's in egg white, right? And vice versa. In the lens cell, you won't have the proteins that will activate albumin expression because that would be terrible. But you do have those which recognize and help activate crystalline gene expression. So this whole process is a very big part of the specificity of gene expression and what cells certain genes get expressed in and which ones they don't. Depends on these. And these production of these proteins is again dependent on what proteins are present in those particular cells, right? And so you get this kind of multi-tiered complex condition of different proteins recognizing different promoters and so on. Now I've gone way over, and I know some of you have lab right after this, so I'm going to shut up for now. We'll continue this on Tuesday. We've got a bunch of stuff left to cover, which is also cool. And for the rest of Tuesday, we'll cover whatever we can of the biotechnology and transgenic stuff.
Tuesday our last class day, according to the schedule, right?
Correct.
Okay.
Good
And then
exam.
Thursday says the final exam, but it's not going to be like a cumulative final exam.
No,
Okay.
it's not cumulative. It's just on, as Ali said, PowerPoint 17 to 20.
Okay, great. I
And
don't
if
have
we,
to go
no, you
study
don't have
way
to go
back.
back. Wow.
Okay.
And it depends on how much time we have on Tuesday to cover that stuff, whether it might not even be PowerPoint 20 or PowerPoint 19. But only what we cover in class is what will be material for what we cover in the exam.
Okay. Awesome. Thank you.
Cool. Have a lovely weekend, everybody.
You too.